Hello, everyone. We have done this a number of times, actually, and you wouldn't believe it. We're just trying to sort out getting our American Sign Language interpreters up on screen, so that's why we've been taking a couple extra minutes here, but hopefully we'll get that sorted soon. Thank you, everyone, for coming to our third session in the President's Dream Colloquium in One Health. My name is Dr. Kaylee Byers. I'm an assistant professor in the Faculty of Health Sciences at Simon Fraser University and a senior scientist in the Pacific Institute on Pathogens, Pandemics, and Society. And before we get going, I do have a little bit of housekeeping. First of all, we have an international audience tonight, and I was told this is one of the most international events SFU has ever had. Is it SFU in general? Yeah, let's say that. So we've got folks from across Canada to Atlanta and Boston to Denmark, Philippines, Germany, Scotland, Italy, Hong Kong, Jordan, Morocco, and Kenya joining us for this. So it's pretty spectacular. We're also offering American Sign Language interpretation for today's talks, and our sign language interpreters are Carly and Liz. They should appear as panelists if you're watching online on your screen, but if you experience any issues accessing the captions or interpreters, please send an email to dream, DREAM, @sfu.ca. To turn your captions on or off online, click the closed caption, the CC live caption icon at the bottom of your window, and you can adjust the size of the font by clicking on the three dots at the bottom of your window that say More and selecting Subtitle Settings. We want to invite everyone online to also contribute questions tonight, so please feel free to share your questions in the Q&A box at the bottom of your screen. We'll get to as many of them as we can. You can also click the like button to upvote questions that you would like asked. So tonight, we are thrilled to invite you to the President's Dream Colloquium in One Health. One Health is a concept that human health isn't just about us. It's also about our shared health with animals and the environment. And it asks us to explore opportunities to support our collective health together, where protecting one protects all. And while this idea of One Health is sort of just recognized in Western science in, maybe the last 50, 60 years, this idea is not new. It's deeply rooted in many Indigenous peoples' ways of knowing and understanding of health. And these knowledges and practices around interconnected health are rooted in the space that we occupy today. And to honor this place, I would like to invite Dr. Mary O'Brien, the Vice Provost and Dean of Graduate Studies, to welcome you all tonight. Mary.
Thanks so much. It's great to see so many of you here today and to know that so many of you are joining us online. I'm new to SFU. I've been here since August 1st, and so I feel so fortunate to be involved in tonight's event. And I also feel incredibly fortunate to be a settler on these lands and to be able to live, work, and play in the unceded traditional territories, including the Squamish, Tsleil-Waututh, and Musqueam nations on which SFU Vancouver is located. I'm just going to say one more thing about the President's Dream Colloquium. I would like to congratulate the team. This provides such a special opportunity for all of us to learn from these world-class researchers, such as Dr. Lipsitch, on impactful and really cross-cutting research. This is such an opportunity, I think, for all of us. So I'd like to thank Dr. Byers and the team and all of you for being here tonight. Thanks so much.
So before I introduce Dr. Marc Lipsitch, I do want to thank everyone who supports this series. We have lots of sponsors, the Pacific Institute on Pathogens, Pandemic Society. We've got numerous groups at Simon Fraser, the Faculty of Science, Faculty of the Environment, Mathematics. We've got CanMod, who's supporting us tonight, both financially and in person. So thank you so much. And please save the date. We've got two upcoming talks. The next talk in our series is Dr. Chris Schell, who's going to be talking about environmental justice and biodiversity on March 28th. He'll be joining us from University of Berkeley. And then following that, we've got Ed Young, who's coming on April 4th. We'll be talking about long COVID. There will be very limited in-person seating for this event, so save the date for when tickets go live. And you can also watch one of the previous talks in our series by Dr. Salome Bukachi, who talked about One Health and the changing global health landscape. Okay. I'm very excited to introduce Dr. Marc Lipsitch to you tonight. I've had the joy of driving around with Marc all day in the car. And when we were first conceiving of this series in One Health, our group at PIPS, we were thinking, who do we want to bring that really looks at different aspects of this connected health? And Dr. Marc Lipsitch made that short list very quickly. Dr. Lipsitch is a world-renowned scientist who has worked in antimicrobial resistance, mathematical modeling of infectious disease transmission, research ethics, to name just a few. And I actually became aware of Marc, not specifically for that research, but actually for science communication. Dr. Lipsitch has done an incredible job communicating science to the public and is somebody who's really passionate about science communication, thinks it's really important. I really value that, and I really want to share my gratitude to you for doing that work. It's not easy, and it's an aspect of science that I think often goes underappreciated or unrecognized altogether, our role in raising awareness about certain issues. And so if I can, and I will because I've got the mic, I'd like to share with you an opening of an opinion piece written by Dr. Lipsitch to showcase some of the science communication skill, because in our President's Dream Colloquium class that goes along with this course, we're also practicing science communication. And I'm going to share an article from the before times, before COVID, in 2014. This is the opening from an article in the New York Times. Officials at the Center for Disease Control and Prevention recently discovered that at least 75 workers there had been exposed to possible anthrax infection. We should be glad that it was only anthrax. Anthrax is a dangerous but non-contagious bacterium. The risk to the exposed workers is real, but there is no danger of transmission to others. Much more troubling would be an accidental exposure to a dangerous, contagious pathogen. And unfortunately, that's also quite possible. That is such a good hook. I would read any academic article that started that way. I mean, at some point I might get really scared and stop reading them, but that's a really good hook. And it's a really important part of science communication, is to draw people in. So I wanted to highlight that. And anthrax is a zoonotic pathogen, so I thought I'd just bring that in, animals and people together. Dr. Lipsitch is a professor of epidemiology at the Harvard T.H. Chan School of Public Health and directs the Center for Communicable Disease Dynamics and the Interdisciplinary Program on Infectious Disease Epidemiology. Tonight, he'll be speaking about virus prospecting in non-human animals and what it can and cannot do as a measure to address infectious disease outbreaks. Please join me in welcoming Dr. Lipsitch. Very good. I'm going to try talking to it again.
It says on.
It does say on.
Oh, no.
How many PhDs does it take to sort out? Right, we can also use this if it ends up being easier, but it should be working. Okay, well, you can have it for show.
Great. Well, thank you very much, Kaylee, for that very generous introduction. Thanks to Simon Fraser and the President's Dream Colloquium, and to Carolyn, Kaylee, and Rakib for organizing this. It's really great to be here, and I appreciate also your accommodating my schedule to make it work out with fewer flights. So I'm excited to talk tonight. It's the first time I've talked about this work, which is the work largely accomplished by an undergraduate named Aishani Atresh, who is a senior undergraduate at Harvard, will be a Rhodes Scholar next year in Oxford, and is someone who I got to know a few years ago and proposed to her about a 3-sentence description of a project. And a couple of years later, this is what she produced with minimal intervention by me. So much credit to her, and this really reflects a lot of digging and thinking that she has done. So I want to start by a couple of words about what I'm not talking about. Probably you can all leave now because these are the interesting things. No, there are a lot of intertwined issues that that people are thinking about very hard right now, including the origins of COVID-19, and something that's been, which I have not spent much time on, and something I have spent much time on, which is work where in the laboratory people are enhancing the virulence and transmissibility of potential pandemic pathogens, or so-called gain of function of concern studies. Those are both important topics. The second of them, not the first, is addressed in this what I think is really good report that I had a role in, but was chaired by the individuals listed here, and came out yesterday. Actually, it was released at the United Nations, and I think is one of the better pieces of work talking about the what's needed in that field. But because that's there, I'm going to talk about something different, which is the question of what we learn and what we don't learn from virus prospecting, which I'll define in a minute. Most pathogens that infect humans eventually, if you go far enough back, came from animals, from non-human animals. The statistics that have been published recently say that 60% of emerging infectious disease outbreaks are caused by zoonotic pathogens, or the pathogens that spill into people from animals. About 70% of which have origins in wildlife, the remainder mostly in domestic animals. You might wonder, where else do we get viruses from? We don't get a lot of Dutch elm disease in people, and that's true of most plant pathogens. So I think that if you look at the list that's in the reference shown, from which the graph is taken there, most of the other ones, if not all, are new drug-resistant forms of existing human pathogens. So if you look far enough back, those probably are also zoonotic. Around 250 different viruses are known to infect humans. There have been estimates ranging from about 100 times that to about 1,000 times that that we don't know, that are in mammals, that some fraction of which could potentially infect humans and are undiscovered. Recently, it's been appreciated more that spillover events are not the only reason why new diseases pop up in humans, or seemingly new outbreaks of diseases. So a really nice paper that was published last year suggested that the Mpox strain, or monkeypox virus strain that was transmitting in mostly gay and bisexual men and other men who have sex with men around the developed world, in a major outbreak in 2022, that probably has been in humans since around 2016, and that's based on signatures of human adaptation that go back in the phylogenetic tree before the widespread that was documented in people in the West. Other suggestions have been that some of the Ebola outbreaks that have looked like they were new in African countries actually were the reappearance of strains that had been circulating in previous outbreaks and had gone latent and then became transmissible again from an infected human that got it many years back. That's more speculative, but is possible. So when we say something is a new outbreak of a zoonotic disease, that doesn't mean that it's a new zoonotic outbreak. It means that it's a new outbreak that might have come from animals, or it might have come most recently from people, and be restarting its transmission, or newly discovered because it gets into a population that's under better surveillance. So the subject of the talk today is virus prospecting, which is an activity that aims to detect viruses that have the potential to spill over from wild animals to humans and cause disease in humans, and trying to detect those before they become a problem in humans, before an outbreak in humans. In the beginning of this century, this became a fashionable thing to do amidst concerns about avian influenza and other possible spillovers from animals to people. And there's been a little bit of work on whether this is beneficial and what the costs are, a little bit on viral diversity and on whether we can use the results of this to predict spillover. And it also has names like viral discovery, virus hunting, or zoonotic surveillance. So it's going out into wild animal populations, studying the viruses that can be isolated from those populations, either by sequencing genetic material if the virus is not viable, or by actually culturing the virus in the laboratory and making copies of that virus. So increasingly, I think people have begun to appreciate the risks of this kind of work. In about a year ago, the Washington Post published a very long article that featured a scientist from Thailand who had been receiving research funding from the US government to go out and study, isolate viruses from wild animals. And this scientist said that he had decided that this was a bad use of funds, that this was risking his field workers' lives and safety because of the inability to protect them adequately, and that it didn't really add much to pandemic preparedness. And he just returned the money to the United States government on the grounds that this was not a good way for him or his colleagues to spend their time. Something kind of similar happened on a larger scale later last year when the United States Agency for International Development, or USAID, canceled a program called Deep Vision. That was a $125 million US program to do virus prospecting. It was a little bit unclear or ambiguous what the reasons for that cancellation were. The BMJ, the British Medical Journal, now called the BMJ, suggested that it was due to a unfavorable risk-benefit profile. But in any case, that was the decision. So if those are partly risk-driven decisions, it's important to ask, what are the benefits of this kind of work? And are we foregoing something important by not doing them? So the questions I want to ask in this talk are, do our ways of looking for viruses in animals identify potential or actual causes of outbreaks? Or do we see them in advance? Do we see them retrospectively? How much has outbreak preparedness depended on our ability to do zoonotic viral discovery in wildlife? In other words, is knowledge of a viral pathogen before an outbreak associated with changes in preparedness, such as the development of countermeasures like drugs and vaccines? And finally, what do we know about the role of novel spillover events in driving major outbreaks of concern as opposed to other sources? So I'll talk a little bit about the background of large-scale surveillance. I'll then zoom into the filoviruses, which is a family of viruses that's most well known for Ebola, but also includes Marburg virus, and these viruses cause hemorrhagic fevers and are obviously well known to the world at least since 2014. And talk about sort of the timeline of what we've learned about those viruses in wild animals and how that's related to our preparedness for outbreaks. Then I'll talk about zooming out into the vaccine development landscape across virus families and which viruses we've prioritized for countermeasure development to answer the question of whether prospecting results have driven research and development for novel countermeasures against known viruses. And finally, I'll talk about health emergencies in humans, answering the question, which viruses are causing major disease events and where. So the US has been a major sponsor, not the only sponsor for sure, but the major sponsor, I think, of viral prospecting studies. And that's through the PREDICT project, which was a $200 million expenditure for the decade of mostly the 2010s. Deep Vision, which was prematurely curtailed in 2023. And the Global Virome Project, which has very large ambitions, has so far been fairly small and is still very much being developed. So the one that we know that we can sort of say the most about because it's been completed is PREDICT. So that program touts as its accomplishments that over 6,800 people were trained for the One Health workforce, that 164,000 animals and people were safely sampled, enhanced laboratory systems, 949 novel viruses detected, 217 known viruses detected, and one of those to date has been demonstrated to be able to infect humans, which was discovered during a human virus outbreak, so not through animal process. So thinking about the filoviruses, these are, as I mentioned, a strand of a family of viruses known for Ebola, for the 4 species of Ebola and Marburg virus. These are very serious infections that cause viral hemorrhagic fever, bleeding from all sorts of orifices, really horrible symptoms, and kill a large number of the people that they infect, depending on the level of treatment available. In just last year alone, Marburg was seen for the first time in 2 countries. And Marburg, for Marburg, we have no approved vaccines or antivirals. And we have a drug that gets used for essentially every new viral infection because it might be helpful, but is not a great option. And then in Ebola, in 2023 alone, there was an outbreak of Sudan Ebola virus in Uganda in humans, causing 142 cases. And that was the first of those that had happened for about 10 years. So this is a timeline of the isolations or of the outbreaks of the 4 types of Ebola viruses. Zaire on the top, the one that caused the big outbreak that everyone knows about from 2014. Thai forest, which caused only 1 small outbreak. Sudan, 3rd, and then a final type of Ebola virus. And so you see that throughout the first ones documented in humans were in the late 70s. There have been increasing numbers of ascertained outbreaks. Probably some of that is because we're looking harder. Some of it may be actual increasing numbers. And the colors suggest whether this is a spillover, a lab accident, or unknown. The shapes of the locations and the size give a sense of the scale, with the 2 very largest ones being shown with these crosses, the 20 else? No. Here is the 2014-15 major outbreak in West Africa. It was the most well-known of these outbreaks. So below are the countermeasures that we have developed. So on the left-hand side are vaccines, different types of vaccines, and monoclonal antibodies and antiviral drugs. And the darker and darker shades are increasing levels of testing, of sophistication of testing, or advanced testing of these countermeasures over the course of several years. And what's remarkable is that essentially all of this started, with the exception of small phase 1 trials of a few, maybe tens of people at most, just for safety. The real development started only in 2015, or 2014, following the outbreak in West Africa. So this large outbreak is what really got us started with R&D. We'd had 40 years of human outbreaks, and nobody was doing much to counter it. when there was a large human outbreak, that started to change. So on this slide, we have the same human outbreaks on the top, and on the bottom are isolations from animals. And these are all isolations of genetic material. So these are not live viruses. No one has found a live virus in an animal, but there have been isolations of genetic material starting in the early, well, mostly in the early 2000s, one a little bit earlier for one of the Ebola viruses. And largely, first of all, we didn't find live virus, and live virus isolation from an animal is required for that animal to be considered the reservoir, as opposed to just an incidental host. And again, these isolations of genetic material happened at a time period when we weren't developing countermeasures, and we continued not to develop countermeasures. So to summarize this experience with Ebola, we still don't really know what the reservoir of Ebola is, at least by the official definition of a reservoir, because no live virus has been isolated. There are many types of bats that show the genetic material and are suspected to be the reservoir. There's a theory that arthropods or insects may play a role. And 2019 was the first time that Ebola was discovered in a bat in West Africa, where most of the outbreaks have been. And that was after the countermeasure development was started and after the largest of the human outbreaks. We have discovered one virus in the Ebola category through zoonotic surveillance prior to an outbreak, but it's never caused a human outbreak and does not seem to be able to cause disease based on studies in the laboratory of trying to get human cells infected with it. And finally, while animals are definitely a reservoir, some animal must be a reservoir, there are these suggestions of undocumented human circulation. So there's studies where individuals who had not been known to have an Ebola virus outbreak infection in the Democratic Republic of Congo from Anne Ramoin's group showed that they did have antibodies that were responsive to Ebola, suggesting that they had probably been infected and not known. As you know, the 2014-15 outbreak included three main countries in West Africa: Guinea, Liberia, and Sierra Leone. And the 2021 reactivation outbreak. So there was another small outbreak in humans in 2021 in Guinea, and that was shown through the sequence of the virus to be related to reactivation of a virus that came from that prior outbreak. That has led this group of authors to suggest that maybe other such reactivations have happened and we didn't know it. Marburg tells a similar story, except that we haven't gotten as far. So on the top is the geographic locations of different isolations of Marburg virus. The first one was in Germany in the late 60s. Humans and other human outbreaks have occurred. And once again, the countermeasure development began in the mid-2000s following significant outbreaks in Central Africa. And if you compare that to the findings in animals, they were late and not linked to human cases. So unlike Ebola, we do know the reservoir or a reservoir for Marburg virus because live virus was isolated in 2009. But most of the cases that we know about are in tourists, and there has not been a clear link of the outbreaks to either epidemiologically or genomically to the presumed reservoir that has been documented. If we zoom out now to think about other viruses, and the first three are what we're calling major viruses, and the rest are less well-known viruses. There are three that were discovered in animals prior to causing human outbreaks. So we do sometimes discover viruses in animals prior to causing human outbreaks. For example, we knew about monkeypox in monkeys in 1958. First human outbreak followed 12 years later. Similarly, Zika virus, which we all heard about a few years ago, was discovered in animals in 47 and humans in 52, and Rift Valley fever. None of these discoveries in animals or in humans, to be fair, initially caused us to do anything in terms of developing new countermeasures. And similar, you've probably never heard of some of these viruses. I had never heard of some of these viruses. And that's because we basically have not been doing much to counteract most of them as well. So you can find animal viruses and then we don't do anything about them, suggesting that the countermeasure argument is not empirically proven. The argument that we develop countermeasures because we find things in animals. That's one direction to look at it, but you can look at it from the other direction, which is we know about a lot of viruses in humans. And if we had countermeasures for all the viruses that we know already infect humans, then you could say we need more candidates. We need to figure out what else to develop countermeasures against. So let's go looking in animals. But as it happens, we have 25 or 26, people disagree about it, families of viruses that are known to infect humans. And families are big things like filoviruses that include Ebola and Marburg, or herpes viruses that include Epstein-Barr virus and herpes simplex viruses and cytomegalovirus and all sorts of others. So these are big groups. And even at this big group level, we have 26 families, of which we have 16 that we have countermeasures for, vaccines mainly, and 10 that we don't. So we have a lot of activities that we could undertake if we are interested in improving our preparedness for human disease, starting with the other 10 families. And moreover, many of these, we have a single, there's a single member of that family for which we have a countermeasure. What's a good example of that? For example, adenoviruses, we have a vaccine for the US military, but otherwise there's not a vaccine, and we don't have it for other members of that. Similarly, for hepatadenovirus is hepatitis B, but that's only one. This is the second half of that table. And so here's another family, the pneumaviridae, which includes the respiratory syncytial virus, and that's the only one for which we have countermeasures. So we have lots of things we could do if our goal is to deal with potential threats to human health. We have at least 10 families and a lot of things within existing families to work on. We are not short of candidates. So this is a table of the viruses that 4 different organizations, the World Health Organization, the US National Institute for Allergy and Infectious Diseases, the Coalition for Epidemic Preparedness Innovations, or CEPI, which has been a real leader in this field, and the UK government have prioritized for countermeasure development. Most of these were discovered during human outbreaks a long time ago, mostly before 2000. Given that we had 20 years before any of these prioritization efforts even happened, and we already knew the sequences, we already knew about these viruses, and we just didn't decide to prioritize them. So it further suggests that we do need to take action, but we don't. But the rate limiting step is not discovery of new viruses in animals. And then the last part of the discussion is to look at what are the diseases, what are the families of viruses and types of viruses that cause the most outbreaks in people? So it's hard to quantify what outbreaks are, but there is a thing called the World Health Organization Disease Outbreak News. So they note outbreaks when they occur. They don't find everything, but they note a number of them. And what you see is that in each region, there's one group of pathogens. So in Africa, it's Ebola viruses. In the Eastern Mediterranean region, it's MERS coronavirus, or Middle East Respiratory Syndrome, influenza A in the Western Pacific. And those account for a majority of the outbreaks. And those are not things that we need to discover or that are unknown. Almost none of these human outbreaks over the period of study are of viruses that were previously unknown or that we need to discover in animals. And if you look at the larger outbreaks, such as the ones that cause public health emergencies of international concern, so that's things like the H1N1 pandemic of 2009, the Ebola outbreak of 2014, Mpox recently, COVID-19, and so forth. So the sort of heavy hitters among the viruses, these are all things that we've known about, and with the exception of COVID-19, which was closely related to something we knew about. So what I would suggest from these different sorts of lines of evidence is first that assessments of spillover in the case of filoviruses, one of our most intense case study in this work, have been inconclusive. It's been hard to find even the zoonotic origins of outbreaks that we know about. So if we think about what's the comparative difficulty of, on the one hand, saying we had an outbreak, let's go look for the source. And on the other hand, saying we might have an outbreak, let's see if we can find the source of that outbreak before it happens. You have a lot more clues in the first case, and we're terrible at that. So it suggests that the second case is going to be even harder. We just haven't found very many viruses, despite looking, that are known to infect humans now that we found before they infected humans in animals. And the corollary of that is that most of the health emergencies have been caused by a subset of known pathogens. The best argument, I think, for virus prospecting is if it would spur us to develop countermeasures. Empirically, that has not happened. What has happened is that we wait until we discover things, we do nothing when there's a lot in animals or in small numbers of people, and then when there's a large enough outbreak in people, we start to develop countermeasures. So it does not seem that the limiting resource for countermeasure development, in terms of urge to act, is discovering animals, nor is it the limiting resource in terms of finding candidates for things to work on, based on the number of viral families that we still don't have countermeasures for. I think there are lots of aspects of this that I didn't have time to talk about in detail, but some of those are that we really don't know, as we've seen on some of those timelines, an increasing frequency of outbreaks. We really don't know if that's an increasing frequency of outbreaks or an increasing ability to ascertain them, or it probably would be both, is my suspicion. I think we did not, the argument I've made does not account for contributions of viral prospecting to basic science. It was really a practical, what are the practical consequences? I think one of the successes of CEPI, the Coalition for Epidemic Preparedness Innovations, is that it was set up after Ebola to try to push the countermeasures to things we know are potential threats, like Ebola, that had been sitting on the shelf for very, very early stage investigations, try to push them to the point where we know that they're safe and we know that for a vaccine, that we know it's safe and we know it engenders an immune response. And to do that for a whole range of pathogens, so that if a new Ebola outbreak or a Marburg outbreak or something like that occurs, we have a countermeasure on the shelf. And if something new comes up that's related to one of those pathogens that's been studied, we can have a head start. And those are things that have little market incentive for companies, but with investment by the public sector can be helpful. And indeed, two of the things that CEPI had invested in prior to the COVID pandemic were vaccines against MERS, which is a relatively close relative of the SARS-CoV-2, and which had the same issues with the development of vaccines, and the mRNA platform that was used by Pfizer and Moderna. They weren't the only ones investing in that, but they had invested in that, and I think it would be interesting to try to quantify the value of those investments in speeding up the world's response. I raised the question of whether when we see a novel spillover event, whether it is a spillover, or when we see a novel outbreak, whether a spillover is a reliable hypothesis, first starting hypothesis, or whether we should always consider the possibility that it's come from a reactivation. And the obvious question arises, what if you're against this, what are you in favor of? And I think that I've talked about the idea of preparing vaccines for known and countermeasures in general for known threats that infect humans, and for general purpose capabilities to fight disease, including the infrastructure to do small scale containment when outbreaks occur. That doesn't solve everything. We've seen, for example, in COVID-19, that test and trace was of partial value, but not complete. But in something like Ebola, it's incredibly important because of the nature of that disease. So I think it suggests that for response and resilience, we should maybe focus a little bit less on sequence and a little bit more on the infrastructure to create new countermeasures rapidly. And some of the ways to do that are to do exactly what CEPI is doing, and colleagues at the World Health Organization and others, to identify high priority pathogens through blueprints, fund clinical trials at the early stages that we can do in the absence of an outbreak, and watching outbreaks in humans. Nigerian scientists and physicians had been warning for years that Mpox was going to be a problem, and that it had acquired the ability to transmit more readily than people knew. And because it hadn't been detected in the West, that didn't happen. So we have lots of sources of really good signals that are at least as reliable as the discovery of a new virus in an animal to spur us to work on pathogens. Other types of research that could pay off are to take the diversity that we have seen already from sampling zoonotic pathogens and human infections, and really try to understand the viral diversity and understand whether we're seeing really new emergences or just branches of existing trees. The dichotomy is often made between surveillance in the humans who are exposed to animals and surveillance in the animals. And I think I would come down, given what I've just said, on a greater focus on endemic pathogens and on the potential to see spillovers through human cases rather than animal surveillance. And finally, stronger local public health systems are always essential to this. So in reflecting on the One Health theme of this series, I guess I've presented kind of a two health view, it seems. And I warned Kaylee that I'm a bit of a two health person myself. I think One Health is a hypothesis rather than a guiding principle for how we should always act. and that we should think about, we should think in the context of One Health of the idea that certainly all of our environmental, animal, and human health and plant health, for that matter, issues are interconnected. But we shouldn't reflexively then jump to doing a particular type of One Health when the evidence points in another direction. So that's the argument I've tried to make today, and I will stop there and take questions. Can I actually put it back on Aishani's face because she did all the work? We did have a lot of discussions, but you can want to talk into this. That one?
Yes, thank you. We can share. It'll be so fun. Thank you for that incredible talk. I get to say that so close to you. Thank you for that incredible talk. Something that struck me was, I often think about the reactivity of our health systems, and that even when we are trying to do a proactive thing, virus prospecting, right, we still get caught in the reactivity of actually responding to it. So I thought that was really interesting. Thanks for sharing all of that. I mentioned at the start that this is also part of a course, a President's Dream Colloquium course. We have 9 students in the class. And in advance of tonight's talk, they read a paper that was related to this talk. So I've got a couple questions from them. We'll turn it over to the audience and then a couple questions online, and then we'll wrap up. I had one question that came sort of the same theme from Nilu, Zaresh, and Jesse. We're sort of talking about RNA viruses, their rapid evolution, the virome. And are there any new promising methods or technologies for improving our detection and how we characterize novel viruses at this human-animal interface?
I don't really know the answer to that because it's not what I do. I just made an argument, at least for the animal side. But I think what the promising new technologies that I can speak to are for developing countermeasures. So the mRNA platform is the best known of those in many places because it played such an important role in COVID. But there are variations on mRNA. There are virus vector vaccines and other types of ways to make vaccines rapidly based on a sequence. We still need to figure out which part of the virus sequence is the most relevant to immunizing people, and figure out what needs to be tweaked. So just using the raw spike protein would not have done it. You have to stabilize it, and that was this consequence of years of work, first on respiratory syncytial virus, and then on coronaviruses that people figured out. So it's not none of these are panaceas, but they all do speed up the process of going from isolating even a sequence, if not even a living virus, all the way to a vaccine more rapidly. And I think the other thing I would say is that that CEPI and others have come up with the notion of countermeasure libraries where you try to develop vaccines against a range of known pathogens. Again, as I mentioned before, as practice essentially as well as if you're lucky, as a way to have something on the shelf that could then be scaled up in production. And if you're not lucky, at least you may be lucky enough to have done something relevant to the virus that does emerge.
Thank you. And so you've already talked about with this question from Sevde was talking about, what do we do with this information? You've already sort of walked us through what this might look like. Are there any zoonotic diseases or emerging zoonotic diseases or novel viral pathogens that you believe warrant particular attention in terms of surveillance and research efforts?
I don't know. It's again something that I don't have a strong view about. But I think these kinds of studies in the human-animal interface and some of these expert committees have actually come up with very good lists, and we should continue to get to work on those as a starting point, and to work on platforms as a hedge against the emergence of something we don't anticipate.
Thank you. Do we have any questions online? Really? Okay. Do we have any questions from the audience? Okay, go for it, Arna. You're gonna have to project.
But in the context of the talk, is it worth looking?
The question was, is it worth it to look at the origins of SARS-CoV-2 for folks online?
Yeah, I think it's worth trying to do. It's going to be very hard in the absence of the data on the early spillover, if it was a spillover from an animal directly. The circumstances of that are still not the evidence about the circumstances of that is not readily available. So I think it will be a hard thing to do. I think we need to keep looking. I think many people are quite pessimistic that we're going to settle the question of where the virus came from, even whether it was zoonotic or not. Yeah. Why is it important? Well, I think that knowing what animals are reservoirs of things that spill over and cause major outbreaks in humans is useful, because we then understand I mean, we have hypotheses for the next one, but and I also think that until we find a zoonotic origin, there will be legitimate questions about whether there is a zoonotic origin. So saying it's an animal is not really good enough.
Arna, I want you to know it was an accident that I accidentally moved to the slide that said what I'm not talking about while answering that question. We had another question at the back.
I think for things that are known spillover risks with potential human-to-human transmission, there's a discussion to be had. Something like Lassa fever or Nipah virus or Ebola virus, that for populations at high risk, there might be an argument for doing that. I think for things that are more speculative, where there hasn't been significant human infection documented or human casualties as a result of infection, it's really hard to make the ethical argument that, especially in the light of real health needs in those same populations from actual pathogens that are spreading. So I think it's hard to make that argument to vaccinate healthy people with a vaccine that may or may not have any purpose.
I'm going to go to another student question, if that's all right. So you pitched us a 2 Health here at the end of our One Health series. So that's really good. But so if we were to actually, so this is a question from Raymond that I'm sort of moving a slightly different direction around. If we were to continue virus prospecting, how might we use One Health to do a better job of that, so that maybe the information we get could be more informative towards action?
Asking a 2 Health guy for One Health
Yeah.
advice.
Yeah.
Well, you're going to get it'll be worth at least what you pay for it. I mean, I think the One Health perspective is that spillovers from animals to humans happen, and that the surveillance in the people that are exposed to animals is more valuable than that. I mean, I think to me, that is the One Health perspective as well as the Two Health perspective. That's all I got to say.
Together. No, that's good. That's good. So again, the students in advance of this talk had read a paper, and one aspect of that paper talked about what you just highlighted, right? Being able to look at the people who are coming into contact with animals, maybe the most in certain situations, folks who are working with animals, maybe hunting animals, consuming wild game. But we know that in Canada, Indigenous peoples and other Indigenous peoples around the world are unfairly targeted by regulations that prohibit traditional practices for reasons of health and safety. So from a policymaking perspective, how can we balance the need to monitor animal and human populations for something like virus spillover with the need to respect traditional cultural practices? And this question is from Monica.
Yeah, I think it's a hard question, and I don't know that I have better answers than anybody else. But what I do think is worth highlighting is, I mentioned this to Kaylee this morning. There's a document from the World Health Organization called The Ethics of Public Health Surveillance. And when I picked up that document, I was expecting it to be about the rights to privacy and to not having your data shared with governments that could use it against you, and considerations like that are very prevalent in many places, and appropriately so. There is a little bit of that in the document, but what the document points out more surprisingly to me, but importantly, is that it doesn't quite go as far as to say there is a right to be under public health surveillance, but it suggests that that it is a duty of governments, at least, to do public health surveillance, which might argue that it is a right of people who are under those governments to have it, because surveillance gives information, and you can fail to deal with a health problem even though you know about it, but you can't deal with a health problem at all if you don't know about it. And so I think, to me, that has a few implications. One is that we should not think of efforts to document and understand infectious transmission in any population as inherently against their interests, or we should try to do it in a respectful way. But also that the value proposition, if you will, of doing that kind of surveillance is that you need to combine it with addressing the problems that you find. And so surveillance and studies and even regulation without action that addresses the problems is just a cost without a benefit. I think trying to make sure that the activities that may be burdensome to people are also bringing information to authorities who can also then provide some value in return is an important piece of it.
Thank you. Charlie, do we have any questions online? Okay, and we'll take one more. Yeah, from here.
Yeah, I mean, your question raises an interesting point, which is not exactly what you were asking, but I'll address that first, and then what you were asking, what I think you were asking. Scientists, for very good reason, try to make the best pitches for why their work is valuable, and we're all in it for good reasons, or mostly all in it for good reasons, and ordinarily, the worst thing that happens if governments, for example, choose to fund science that doesn't work out is that some money gets spent that could have been spent maybe doing something else. There is a small subset of research including some types of viral prospecting and some of the kinds of studies that I talked about with viral enhancement, where the risk is actually a completely different type of risk. It's a risk of causing an outbreak or a pandemic in the worst case. And that type of research needs a very different level of scrutiny, because wasting money is one thing, but causing a problem is another. And that one thing I like about this report is that it delineates 3 categories of studies that need special scrutiny: those with known pathogens, known potentially pandemic pathogens, such as smallpox virus, which is extremely heavily regulated; those with enhancement of existing pathogens that are not pandemic capable, but could become so in the course of the experiment; and thirdly, viruses with unknown risks, but that are taxonomically related to ones that are thought to be high risk. And the virus prospecting would probably fall in that third category. So I think we do need to bring those kinds of thinking together. But you asked a different question, which is, what can we do to prevent the next pandemic? I mean, I think preventing a pandemic from occurring is a very, very tall order, given the challenges of containing an outbreak of a highly transmissible virus. I don't think, despite what some people were saying, that there was ever any realistic chance, once Wuhan was full of cases, that the world would not be full of cases soon thereafter. And so I think preventing a pandemic full stop is an admirable goal, and that things like surveillance at the human-animal interface and rapid response to small outbreaks, such as, I mean, not small in this case, but to smaller outbreaks like the Ebola one outbreak in West Africa, is a critical part of that. But I think we also really have to be faster at developing countermeasures and at setting up public health responses. And doing that, I mean, there's a weakest link problem in infectious diseases that's not true with everything else. That's why some of us were saying from very early on that Wuhan was the harbinger of global spread, because it doesn't matter if every country except one does a good job, because as soon as those countries stop doing a good job and some other country has not done a good job, they're at risk again. So I think keeping containment when small is the only option. And we have to try that, but we also have to be able to scale up countermeasures in the event we fail.
Thank you. I think we'll end it there. Please, another round of applause for Dr. Marc Lipsitch. Thank you so much. Okay. We are going to wrap up. I do want to thank again all of our sponsors for tonight and this whole series: Simon Fraser, CanMod, the Pacific Institute. Thank you for your contributions. I also want to thank again our American Sign Language interpreters online, Carly and Liz. Thank you so much for being with us here tonight. A reminder that we do have 2 more talks in this series coming up. Please join us for Dr. Chris Schell's talk on March 28th, and keep your eyes peeled for for Ed Young's talk on April 4th. And that's the end of the slides. This series wouldn't be possible without our small but mighty team. So big thank you to Charity, Stacy, Gladys, and Rakib, who are outside. And thank all of you for coming. See you at the next one.